Alright, next we're going to look at the processes of muscle metabolism like direct phosphorylation, anaerobic glycolysis, and aerobic respiration. Here you can see an image that shows you those three processes. You have direct phosphorylation, which takes a phosphate from creatine phosphate, adds it to ADP to generate creatine and ATP. This gets us 1 ATP per creatine phosphate, and it only takes about 15 seconds. So if we need ATP quick, this is one method. Muscle cells actually store about 2 to 3 times of the creatine phosphate compared to how much ATP they store. So it's a useful method for generating ATP quickly. You also have the anaerobic mechanism, which utilizes glycolysis and fermentation. This process produces lactic acid as a byproduct, but does give us 2 ATP per glucose molecule, with glucose being the primary energy source, and it's pretty quick, about 30 to 40 seconds. But the method, I should say, that makes the most ATP per energy source is going to be the aerobic mechanism that utilizes oxygen, or it's also known as aerobic cellular respiration. Now, its energy source is generally glucose, but it can be pyruvic acid, free fatty acids from adipose tissue, or amino acids from protein catabolism. Oxygen use is required in the aerobic mechanism and not required in direct phosphorylation or the anaerobic mechanism. But it gives us anywhere from 32 to 34 ATP per glucose and generates CO2 and water as a byproduct. Now, the downside is it takes hours. So sometimes muscles need ATP quicker. We might need to regenerate ATP while the metabolic pathways adjust to sudden higher demands, such as during increased vigorous exercise. Creatine phosphate is going to be a great resource for exactly that reason. Again, it produces 1 ATP per creatine phosphate and takes 15 seconds. The anaerobic glycolysis method might be great when muscle contractile activity reaches 70% maximum because bulging muscles compress blood vessels. That would impair both oxygen delivery and it would prevent pyruvic acid, which can be converted into lactic acid, from exiting the muscles. So that anaerobic glycolysis process gives us just a little bit more ATP than the direct phosphorylation, and both creatine phosphate and glucose, when used in either direct phosphorylation or the anaerobic mechanism, can generate ATP by some type of product catabolism in order to get us ATP for the muscle. With the anaerobic glycolysis, the initial phase glycolysis is the same process that occurs during the aerobic pathway, but without oxygen, this process can't proceed past glycolysis. Instead, it just uses the glucose to produce 2 pyruvic acid molecules and 2 ATP per glucose, and it's pretty quick. It takes 30 to 40 seconds. The lactic acid produced by the anaerobic mechanism, though, does diffuse into the bloodstream and usually goes to the liver or the kidneys or the heart and gets broken down, basically converted back into pyruvic acid. But sometimes it gets stuck in muscles and it can contribute to muscle soreness, but it is not the sole cause. And this process, the anaerobic glycolysis process, is 2.5 times faster than the aerobic pathway, but it does produce only 5% as much as ATP. So it's partially responsible for sore muscles after exercise, but it is another way to get us ATP, but it's not the best. The most efficient way to get ATP, even though it takes hours, is the aerobic pathway, where we use glycolysis and other processes that require oxygen, and this occurs in the mitochondria. It does include glycolysis, but also other steps. It produces anywhere from 32 to 34 net ATP, depending on your textbook, and has byproducts of carbon dioxide and water. Again, that downside, though, is it takes hours. So the most efficient mechanism of muscle metabolism is definitely the aerobic mechanism, or aerobic cellular respiration, that utilizes oxygen. But if you need ready available ATP, direct phosphorylation and anaerobic mechanism are a little bit quicker. There's also this idea of muscle fatigue that occurs when the muscle organ is in a state of physiological inability to contract. So the muscle can't contract because it's tired. This could occur when we can't produce enough ATP, especially with how quickly we're trying to continuously use a muscle. If there's a deficit of ATP, that would result in kind of either weird contractures or no contracture of a muscle. We could also see muscle fatigue when lactic acid accumulates in the muscle and if there's any ionic imbalances. So something that might cause this muscle fatigue is vigorous exercise. It definitely causes dramatic changes in muscle chemistry. And for a muscle that's fatigued to return to a resting state where it's ready to contract again, oxygen reserves in myoglobin have to be replenished, lactic acid has to be converted into pyruvic acid and kind of moved out of the muscle, glycogen stores must be replaced so we can break down glycogen to glucose and power the aerobic mechanism, and ATP and creatine phosphate reserves, even though it's not a bunch, must be resynthesized. We used to call these kind of parameters for a muscle to return to its resting state the oxygen debt. But now it's called excess post-exercise oxygen consumption, or EPOC or EPOC. It's just the extra amount of oxygen that's needed for all the restorative processes for a muscle to return to its resting state. And when we're talking about a muscle returning to its resting state, it's going to be able to return to the ability of contraction. And when it comes to the force of muscle contraction, there's different things that affect that force, such as the number of muscle fibers contracting. The more muscle fibers in a muscle, the stronger the contraction. The relative size of the muscle also affects the muscle force. The bulkier the muscle, the greater its strength. Also the frequency of stimulation, because the more rapidly a muscle is stimulated, the greater the force it exerts. And last but not least, for the force of contraction, it is also affected by the degree of muscle stretch. Muscles contract actually strongest when the muscle fibers are anywhere from 80 to 120% of the normal resting length. That means that if a muscle's already contracted, it's not going to contract with much force. If a muscle's slightly contracted, it's still not going to contract with as much force as if it was extended or at rest. So that's definitely going to affect the force of contraction. And then one of the last things we're going to talk about is muscle fiber types. So we're going to be looking at functional characteristics like speed of contraction. Some muscle fibers are going to contract using really fast ATPases that break down ATP quickly. So that gives us slow and fast fibers. Others are going to be differentiated by ATP forming pathways. Some utilize the aerobic pathways primarily, so they're considered oxidative fibers. Others use the anaerobic glycolysis more, and they're considered glycolytic fibers. So if we combine these criteria, they actually give us 3 categories: slow oxidative, fast oxidative, and fast glycolytic fibers. So slow oxidative fibers have slow ATPases, they're fatigue resistant, and they use the aerobic mechanism, so they depend on oxygen. We see a lot of these fibers in marathon runners. They have more skeletal muscle fibers that are converted into this type. You also have fast oxidative fibers, which also require or depend on oxygen. They contract quickly as opposed to the slow oxidative fibers because they have fast ATPases, and they have moderate resistance to fatigue. And then you have fast glycolytic fibers, which do not depend on oxygen, but do contract quickly because they have fast ATPases. But they are easily fatigued, so they're best in sprinters, and sprinters tend to have more skeletal muscle fibers converted into this type. And before we leave muscle, as far as Chapter 9 is concerned, we're going to look at smooth muscle to compare it to skeletal muscle. Smooth muscle is composed of spindle-shaped cells that are thicker in the middle and thinner on the sides, and they truly do look like fibers. They have a diameter of 2 to 10 micrometers in length and several hundred micrometers in an organ. They lack the coarse connective tissue sheaths of skeletal muscle, but they do have an endomysium. They're usually organized into 2 layers, a longitudinal and circular layer, with the orientation of the muscle fibers opposing each other in a perpendicular fashion. Where are they found? They're found in the walls of hollow organs, like the bladder. They're also not found in the heart. They have essentially the same contractile mechanisms as skeletal muscle, with some modifications. Here we can see an image where we're seeing a look at the small intestine smooth muscle and the arrangement of the 2 layers, both longitudinal and circular, of smooth muscle in the walls of the intestines, which is a hollow organ. In a hollow organ, the longitudinal layer of the smooth muscle contracts, and the hollow organ then dilates and shortens. Where with the circular layer, when it contracts, the organ's lumen constricts and elongates. It's this back and forth dilation and shortening, constricting and elongating, that squeezes foodstuffs, chyme, waste, and other things through hollow organs. This establishes a back and forth contraction and relaxation phase that we see in smooth muscles called peristalsis, which is mostly meant to squeeze substances through the lumen of hollow organs, but does a little bit of mixing. And when it comes to smooth muscle, it's not innervated by somatic nervous system neurons. Instead, it's innervated by the autonomic nervous system, or neurons belonging to the autonomic division of the nervous system. It uses hormones like epinephrine and norepinephrine, and does very much respond to local chemical changes such as excess carbon dioxide or an acidic pH. Smooth muscle also lacks neuromuscular junctions, and we don't see neuromuscular junctions in smooth muscle. Instead, we see innervating connections that have bulbous swellings, and these are called varicosities. The varicosities release neurotransmitters like the axon terminals of somatic motor neurons with skeletal muscle, but they release them into really wide spaces, which are synaptic clefts, but they're so wide we actually call them just diffuse junctions. Here you can see what would be an axon terminal in a somatic motor neuron, and in this autonomic neuron is really just bulbous swellings at the end of the axon. Those are the varicosities that contain synaptic vesicles and neurotransmitters like epinephrine and norepinephrine. They go along the different layers of smooth muscle, and they're going to have very diffuse junctions or really large spaces in comparison to the neuromuscular junction called diffuse junctions. Other differences between smooth muscle and skeletal muscle include that the sarcoplasmic reticulum in smooth muscle is less developed, and it's going to lack a specific pattern. Because T-tubules are absent, we also just don't see the organization that's present in skeletal muscle in smooth muscle. We do see something in smooth muscle that we don't see in skeletal muscle, which is the plasma membranes have these pouch-like infoldings or areas on the outside of the sarcolemma called calveoli. And in these calveoli, there is calcium. It's sequestered in this extracellular space near the calveoli, and that's going to allow for a rapid influx of not only sodium, but calcium when ion channels open upon stimulation. There's also no visible striations, no sarcomeres, but we do have actin and myosin myofilaments still present in smooth muscle. So with actin and myosin, we still get increased overlap that leads to contraction. But they're not arranged in a regular form to produce A or dark bands and I or light bands. Instead, the thick and thin filaments, or the myosin and actin myofilaments, are arranged diagonally, and that actually causes smooth muscle cells to contract in a corkscrew manner. And these actin and myosin myofilaments are going to be attached in places called dense bodies, which are similar to the Z-disc at the ends of sarcomeres in skeletal muscle. These dense bodies are arranged in regular intervals, but it does not create striations. Also keep in mind, for smooth muscle, there is absolutely no troponin-tropomyosin complex. Here is a skeletal muscle at the top relaxed. Here's a smooth muscle at the top relaxed, and you can contrast it with skeletal muscle. And here at the bottom you have the contracted smooth muscle cell. You can see the spindle shape in the top image, which is thicker in the middle and thinner on the ends. You can also see the calveoli, or those infoldings along the sarcolemma for the smooth muscle cell. Then you have the contracted smooth muscle cell, and it is just kind of squished and dense compared to the relaxed smooth muscle cell. So when we're talking about smooth muscle as an organ, it's whole sheets of smooth muscle that exhibit contraction. It's a lot slower than skeletal muscle, but these sheets do contract in unison because there are gap junctions between adjacent smooth muscle cells, which allows for very quick communication between cells. And once that contraction in a sheet of smooth muscle is initiated, the action potentials are easily transmitted from one cell to another. Some smooth muscle cells actually act as pacemakers and can set their own contractile pace for whole sheets of smooth muscle. So they share kind of that self-excitatory or automaticity that we see in some cardiac muscle cells. And they're able to depolarize without external stimuli. But keep in mind that the contraction mechanism is really similar. In order for smooth muscle to contract, we still need actin and myosin to interact and form a cross bridge to initiate that sliding filament mechanism. Calcium is still the final trigger for contraction in smooth muscle and in cardiac muscle, and as always in skeletal muscle. Calcium is also still released from the sarcoplasmic reticulum, but also from the extracellular space, and it gets to enter that sarcoplasm. And since there's no troponin-tropomyosin complex, in smooth muscle, calcium binds to calmodulin and activates it. And it's calmodulin that activates another enzyme called a kinase enzyme, which transfers its phosphate from ATP to myosin cross bridges. and that's going to lead to the phosphorylated cross bridges interacting and the actin and myosin overlap that produces sarcomere shortening. And then smooth muscle is just going to relax when the phosphorylation decreases and the intracellular calcium levels drop as the calcium is actively pumped back into the sarcoplasmic reticulum and out of the cell back into those calveoli. So pretty similar to skeletal muscle, except for the difference between having and not having striations, having troponin and tropomyosin versus calmodulin, and then the general structure of where we find it and how it's oriented. But as far as there is something similar to a neuromuscular junction, the excitation contraction, coupling and a cross bridge in smooth muscle, that stays the same for skeletal muscle, with just some slight variations. All right, and that is it for the chapter all about muscle. Make sure to review and make sure you understand all the parts. It's a lot of terminology that was introduced, so that's something that I would focus on understanding.